Hello everyone. I guess you must be busy with your last minute revision for your GCSE papers. Here are a few questions which can help you score well in your GCSE Biology Paper 1. So let's see these questions. Question 1. A student observes a plant cell using a light microscope. The student measures the length of the cell in the image as 15 mm and the actual length of the cell is 0.005 mm. We have to calculate the magnification. So let's see how we're gonna do it. We have magnification as the image size and we'll divide it by the actual size. Over here, the image size is 15 and we'll divide it by 0.005 so that we get 3000. You get 1 mark for the actual calculation and 1 mark for the correct unit, so do not forget the unit. Next, give two differences between a light microscope and a scanning electron microscope. So we can take any two forms. The light microscope uses light and electron microscope uses electrons. Light microscope has lower resolution and electron microscope has a higher resolution. We can take other points as light microscope has lower magnification whereas electron microscope has higher magnification. Light microscope can view living specimens whereas electron microscope cannot. So you get 1 mark each for the correct point of difference. Next, question number 3rd or question C. Explain why living specimens cannot be viewed with an electron microscope. So this is a 3-mark question, therefore you need to have 3 valid points or the 3 keywords as per the marking scheme. The electron microscope requires a vacuum, so 1 mark for vacuum. The other one, the living specimen cannot survive in a vacuum, so the living specimens can't survive in vacuum. And the 3rd point or the 3rd mark is for that these specimens must be dead and dehydrated to be viewed. So dead and dehydrated. Okay, let's move on with the next part, next question. Question 2 deals with osmosis. A student investigates how different concentrations of salt solution affect the mass of a potato chip. The first question is identify the independent variable and the dependent variable in this investigation. So the independent variable is the concentration of the salt, whereas the dependent variable will be the percentage change in the mass of potato chips. Independent variable is the salt solution because the question is asking how different concentration of salt solutions affects the mass of potato chips. So the change or the dependent variable will be seen in mass of potato chips. Next question. The student calculates percentage change in mass rather than just the change in mass. Suggest why. Again, it's a 2-mark question, so the answer must include 2 valid points. Percentage change allows comparison between chips of different starting masses, and the results are standardized and not affected by initial size differences. Question C. A potato chip placed in distilled water increases in mass. Use the term water potential to explain why. Now in this question, it is for 2 marks again. We have to use the keyword or the, as per the marking scheme, which is required over here, the term water potential to explain this. So we'll say that distilled water has a higher water potential than the potato cells. Therefore, water moves into the potato cells by osmosis and it is down across a water potential gradient. So you need to mention 2 points about distilled water having higher water potential than potato cells and the results are standardized, and due to this, water moves into the potato cells by osmosis, that is down the concentration or down a water potential gradient. Water potential gradient. Alright, let's move to the next question. So the third question is related with the heart and circulatory system. Question A. What is the name of blood vessels that carries oxygen-rich blood from the lungs to the heart's left atrium? It's a 1 mark question, direct question. So the answer for it is pulmonary vein. Remember always, pulmonary vein is the only vein that carries oxygen-rich blood because it's moving from the lungs to the heart, to the left atrium of the heart precisely. Second, explain one reason why the left ventricle has a thicker muscular wall than the right ventricle. Again, it's a 2 mark question, so we'll give 2 strong reasons. First of all, the left ventricle pumps the blood to the whole of body, except lungs. Therefore, it requires higher pressure. The second answer, the second part of this answer is the right ventricle only pumps blood to the lungs. The right ventricle only pumps the blood to the lungs, and therefore it's a shorter distance and lower pressure. All right, let's see the next one. What is the job of the valves inside the heart? It's a 2 mark question, so we'll answer it precisely. And valves, we know that it prevents the backflow of the blood, and they ensure that blood flows in one direction. So 2 valid points for this answer for 2 marks. First, it prevents the backflow of the blood and makes sure that it flows in one direction. Let's see another question. Question number 4. A pupil investigates how pH affects how quickly amylase breaks down starch. They test pH values for 6, 7, 8, and 10. Describe a step-by-step method for this investigation, and you must mention concentration of amylase used, how you measure reaction rate, what iodine solution is for, and B part says that at pH 4, the iodine solution stayed blue-black. Explain why. So first, A part is of 4 marks. And we have to describe the step-by-step method. Also, we must mention these things which are given over here in our answer. So we'll add the same concentration and volume of amylase to a test tube. Same concentration and volume of amylase. Example can be 2 cubic centimeter of 1% amylase. Now we'll add the same volume of starch solution. The buffer solutions are used to maintain different pH values like 4, 6, 7, 8, and 10, precisely given over here. At regular intervals, let's say every 10 seconds, we remove a drop of mixture and add to a drop of iodine solution on a spotting tile. Then, we measure the time taken for iodine to stop turning blue-black. That is when the starch is fully broken down. And we'll repeat at each pH for the reliability. Now this is a complete 4 marks answer, and it is logical also with a complete method. At pH 4, the iodine solution stayed blue-black. Explain why. At pH 4 precisely, the amylase is denatured. It's a 2 mark question again. You'll get 1 mark for the denatured word over here, and then the rest for the explanation. If it's denatured, it cannot break down starch. So starch remains and the iodine turns blue black. Question 5. A quite easy topic we can say, but an important one for scoring. Give one example of a disease caused by a virus and one example of the disease caused by bacteria. So you can name any. We have viral diseases like influenza, HIV, measles, TMV, which is the plant one. So we have influenza, HIV, measles, and TMV for plants. Explain why antibiotic drugs work against bacterial infection but not against viral infections. Oh, just a minute. So, for bacterial disease, we have salmonella, tuberculosis, we have strep throat, and many other. Anyone you write and you can score 1 mark. Now let's come to part B. So what happens over here that antibiotics kill bacteria by disrupting the cell wall synthesis or specifically protein production. And the viruses, they replicate inside the host cell. So antibiotics cannot reach them without damaging the host cell. Question 6 says, describe how being vaccinated leads to immunity against a pathogen. What is a pathogen? A disease causing microorganism is a pathogen. Alright. So vaccines contain the dead or weakened pathogen. This is the key word, dead or weakened pathogen, or you can say the antigen. This is the first point. Next, the white blood cells or the lymphocytes, they produce antibodies specific to the antigen. Specific to antigen. Therefore, the third point is the memory cells remains in a body, allowing a faster and stronger response on future infection. 3 marks and 3 valid points. Question B. Explain how a high vaccination rate in a population can protect people who are not vaccinated. The high vaccination rate means what? Fewer people can catch and spread the pathogen. So this reduces the chance of unvaccinated individuals encountering the pathogen, and it is the herd immunity concept. A student uses Pond to investigate how light intensity affects photosynthesis rate. They count oxygen bubbles per minute. What is the independent variable in this experiment? The independent variable, of course, is the light intensity, because again, the question is how light intensity affects the photosynthesis rate. Explain why the student keeps the water temperature the same throughout. Temperature is the limiting factor for photosynthesis. Temperature is the limiting factor for photosynthesis. Therefore, if temperature changes, it becomes a confounding variable, and the effect of light intensity cannot be isolated. Cannot be isolated. So you have to explain the relationship between the change in temperature and how will it affect the light intensity and thus photosynthesis rate. At very high light intensity, the bubble rate stops increasing. Suggest 2 possible limiting factors for this. Any 2 you can write. You can take up the carbon dioxide concentration, the CO2 concentration, the temperature. If it's too high, what will happen? It will cause the enzyme denaturing. The amount of chlorophyll which is there, or the leaf health. Write a word equation for photosynthesis. That's a very easy part. You must know this and you should write this. We have carbon dioxide plus water, and it gives us glucose plus oxygen. Question number 8 has a table which shows the results from an experiment measuring the rate of photosynthesis at different temperatures. Question A, calculate the mean value of Y. Over here, the value Y. How we'll calculate the mean? We'll sum these up. 12.4 plus 13.1 plus 12.9, sum of the observations by number of observations 1, 2 and 3. So if you add it up, you'll get 38.4 divided by 3, which is 12.8. Do not forget the unit. I'm telling again, cubic centimeter per hour is the unit. Next, identify the anomalous result in the table and circle it. So if we observe temperature 30 degrees Celsius, the value 50.2 is much higher compared to all the values over here. So careful observation for this is necessary, and it is much higher than 42 or 41.5. Therefore, this is the anomalous result in the table. Suggest one possible reason for the anomalous result. The possible reasons can be measurement error, a sudden change in light intensity, or the faulty apparatus. Any one will be acceptable. Next, state one variable that should be kept constant in this investigation. Light intensity should be in control, CO2 concentration, carbon dioxide concentration should be in control, and volume of the water or the plant which is used should be in control. Question 9. Aerobic versus anaerobic respiration. Write a balanced chemical equation for aerobic respiration. It mentions a balanced chemical equation. C6H12O6, this is glucose, plus 6 molecules of oxygen gives 6 molecules of carbon dioxide plus 6 molecules of H2O. Remember, if you just write the equation and do not balance it with the right number of molecules, you might lose 1 mark. B. Give 2 ways in which aerobic respiration differs from anaerobic respiration in humans. You can say any 2 forms about it. Aerobic respiration requires oxygen, anaerobic doesn't. Aerobic produces around 36 to 38 ATPs. And anaerobic produces 2 ATP. Aerobic produces CO2 and water, whereas anaerobic produces lactic acid in humans. Aerobic occurs in mitochondria, whereas anaerobic respiration occurs in cytoplasm. The location can be a valid point of difference. Next, during intense exercise, leg muscles starts to feel tired and sore. What causes this? Precisely during intense exercise, our muscles perform anaerobic respiration. So anaerobic respiration results in formation of or production of lactic acid in humans. Therefore, this lactic acid buildup causes tiredness or soreness. Oxygen debt. After running a 400 meters race, an athlete breathes heavily for several minutes. What is meant by the term oxygen debt? This is a very easy question, direct to the point. Oxygen debt. What is oxygen debt? It is the amount of oxygen needed after exercise. Needed after exercise. For what? To break down lactic acid buildup during anaerobic respiration. So you have to explain the whole term, and 2 marks, 1 marks for needed after exercise and the next one for what. Explain why athletes' breathing rates stay high even after stopping. Again, it's a 2-mark question, so we'll explain it in 2 points. The extra oxygen which is used to convert lactic acid back into glucose or to repay the oxygen debt. This requires continued deep and fast breathing to get oxygen into blood. Get oxygen into blood. The first part, will be explanation of oxygen debt, and then we need more oxygen to get oxygen back into the blood. That's all for the video. Hope these questions really help you do well in your exams. All the best and perform well.